Okay, so in previous video we have learned about missense mutation, where one of the amino acid is changed due to the codon coded differently. Okay, so it only affects one amino acid. Now, nonsense mutation, it occurs when base substitution results in changing a codon. Okay, it also changed one codon, but change into a stop codon, which will cause the translation to be terminated prematurely. So, stop codons such as UAA, UAG, and UGA. So the earlier the gene sequence that this mutation occurs, the more truncated. Truncated means the polypeptide chain becomes shorter. So now the polypeptide or the product, more likely it will be unable to function. So example, initial our normal DNA is ACC, it will be coded UGG, okay, in the transcription process. So UGG is coded for tryptophan. So this is very normal DNA. Now after the base substitution occur, the T replace the C. Now it will be transcribed as UGA instead. So UGA become a stop codon. So during the translation, when the stop codon enter the A site of the ribosome, this amino acid chain will stop to grow. So our polypeptide chain will become truncated, which is become shorter. Okay, so this is example of nonsense mutation. This is our normal DNA. It will be coded for normal amino acid like this. So now this T replace the C in the original DNA. Okay, so initially this is our 5' to 3'. So our template, DNA template is GTC. So it will be transcribed into CAG. Okay, CAG code for glycine. So now TAG becomes ATC. So our mRNA based on the template 3' to 5' of DNA becomes UAG. Okay, so UAG is a stop codon. So now this amino acid will become shorter. Okay, halfway it will stop. So the rest of the amino acid will not be coded anymore. So the difference between missense mutation and nonsense mutation. For missense mutation, only one amino acid is changed. The other is still the same. While nonsense mutation, halfway truncated. So suddenly it stops in the middle. So the rest of the amino acid will not be coded anymore. Okay, silent mutation. When base sequence has changed, it will transform one codon to another codon that will still code or translate to the same amino acid. So it is called silent because it does not change our amino acid product. So there is no effect on the protein. Okay, due to we have redundancy in the genetic code table. So many different codon code for the same amino acid. So example, normal DNA is TCG. After transcription, it becomes AGC. AGC code for serine. After base substitution, A replace G. TCA transcribed into AGU. If you refer to genetic code table, AGC and AGU both also code for serine. So after the mutation occur, the amino acid is still the same. So it is like a silent process. Okay, so this is the example of silent mutation. GGC code for glycine. GGU also code for glycine. So it has no effect on the amino acid sequence. So the summary for all the three types of base substitution. This is silent mutation, this is missense, this is nonsense. Okay, so the next type of the gene mutation is base insertion. One or more nucleotides are inserted in the DNA, which results in frameshift mutation. Okay, example, original DNA, original mRNA, original amino acid sequence. Okay, because codon will be read every 3 nitrogenous base. Okay, you read as CTC, CAA, GGA. So instead of TTT, now it will be read as CTT because of one base is inserted. So the TTT, this T will be shifted behind. Okay, so now it becomes TGG. So due to the codon is different, okay, our amino acid also is different. So instead of lysine, now it becomes glutamic acid. And proline become threonine. Base deletion is one or more nucleotide disappear, removed or deleted in the DNA, result in frameshift mutation. So example, original was GGA, TTT, GGA. So now one T is deleted, removed. So the behind nucleotide will be pull forward. So now GGA become TTG because we always read every 3 nitrogenous base. So TTG will be transcribed into AAC, and AAC will code for asparagine instead of lysine in the original nitrogenous base. Okay, so both the base insertion and base deletion will cause frameshift mutation. So what means frameshift mutation? It is changing the reading frame. The reading frame is the grouping of the codon. Codon is grouped every 3 nitrogenous base during the translation. So in the gene mutation, both the base insertion and base deletion will change the mRNA strand. Okay, because it changed the DNA sequence, changed the mRNA strand, so therefore it changed the codon and it changed the amino acid sequence. So the gene will be transcribed wrongly, okay, transcribed in the wrong 3 codon. So codon will be translated differently and it will abrupt the coding sequence of amino acid. Thus, codon will be coded different amino acid. So this will change all the amino acid at and after the point of mutation, starting in the middle of the codon. So originally the stop codon will not be read or may be created at an earlier site. But due to frameshift mutation, the protein produced may be abnormally short, abnormally long, or they contain the wrong amino acid, which is most likely not functional. So the effect of frameshift mutation is very serious, severe, and harmful to human. It is most likely lethal. Okay, which cause death to human. Example is thalassemia. So summary for frameshift mutation caused by base deletion and base insertion. So normally our normal DNA, CTC, CAA, GGA, TTT, GGA. So for base deletion, one of the base, nitrogenous base, is deleted here. Okay, this one. So instead of reading as CAA, now this C is deleted. Now it becomes AAG. Okay, it will be pulled forward. Then become GAT, TTG, and GA behind. Okay, so becomes glutamic acid, serine, leucine, asparagine, leucine. So all these amino acid sequence has changed. While base insertion, okay, let's see. C is inserted here. So C is inserted here. The TTT will be pushed backward. Okay, so become CTT, TGG. So it changed the amino acid sequence. So why base insertion and base deletion is more lethal rather than base substitution? Because mRNA is read as a series of nucleotide triplets during translation. We read every 3 nitrogenous base. So insertion and deletion of nucleotide will alter the reading frame or coding system of the genetic message at mRNA, which causes the frameshift mutation. So the gene to be read is in the wrong 3 base groups starting from the point of mutation. Change in codon, change in amino acid, different polypeptide produced, different protein produced, loss of enzyme activity, or a non-functional protein is produced. Okay, as a summary, initially we read as the fat cat ate the rat. Okay, which we understand the meaning here. So if substitution here becomes the fat car ate the rat. So it has different meaning. While base insertion, the fat cat ate rat. So it has no meaning now. Base deletion, we delete the F here. So it has totally lost the meaning here. So both these cause frameshift mutation. While base inversion, instead of rat, it becomes star. It rotate 180 degree. Okay.